Hi, Gen Bio. Today we are going to be talking about the nervous system and especially focusing on sensory physiology. So, sensory input from the world around us and how it is processed by our nervous system. Okay. So, the first slide here is showing you a picture of the brain. It's a diagram, not the actual specimen. So I just want you to orient yourself first. So your eyes would be on this side. We're looking kind of from below. Okay. So this is like from if you were looking up through your chin. Okay. This is a bottom up view. Okay. So again, this is the front. Your eyes would be here. This is the back of your head over here. And this is the brainstem leading to the spinal cord. Okay. So now that we're oriented, I'm going to point out that there are 12 cranial nerves. And these cranial nerves are important because we want you to understand that they are responsible for processing sensory information from the world around you, as well as responding. Okay, so some of them are sensory nerves that allow us to take in sensory input and allows our central nervous system to process that. And then some of them are motor nerves, which allow us to respond to a stimulus. And then additionally, some of them are mixed. So some have fibers that are running towards the central nervous system as well as fibers that are running away from the nervous system. So you'll hear me describe some of them as sensory nerves and some of them as motor nerves and some of them as mixed. Okay. But there are 12 total, and they are identified by both a Roman numeral and a word that describes their function. Okay, so for instance, cranial nerve 1 is also known as the olfactory nerve. Okay, and as you might guess, it has something to do with our sense of smell. Cranial nerve 1 is a sensory nerve, and so it is taking input from chemicals that are in the air, odorants. Okay, and it is allowing us to process that information. Okay, so it's taking in information about odorants that are in our environment. Okay, cranial nerve 2 is also a sensory nerve. That is the optic nerve. Okay, so the optic nerve, again, as you might guess from its name, is referring to what we can see. Okay, so it's a sensory nerve and it's taking in visual input. Okay, nerve 3 is the oculomotor nerve. And that is a motor nerve, again, as you can probably guess from the name. And it's responsible for some movement of muscles that allow the eye to function. So some of them allow the eye to move, and some of them control the muscles that allow um, more or less light into the eye by controlling the size of the pupil. Okay. And then I'm going to just read you the rest of them. So cranial nerve 4 is the trochlear nerve, and I'll point out for the first 6 here where they are on the brain. Okay, so we looked at cranial nerve 1, cranial nerve 2 would be entering here. Okay, so it actually would be connected to the eye, it's the optic nerve, and then it crosses at this little X here, which is known as an optic chiasm. Okay, and then it's going to take that information back to the back of the brain where visual input is actually processed. Okay, and then cranial nerve 3 is this little one here. It's kind of on the midline also. That is, like I said, that's the oculomotor nerve, nerve 3. The trochlear nerve is nerve 4, and it comes out a little bit more on the sides. Okay, so it's a smaller one, and it's located a little bit more lateral than the first 3 nerves. The trigeminal nerve is nerve 5. That's a much larger one, and it's on the sides as well. And then nerve 6 is the abducens, and that's more on the midline again. That's right here. Okay. And then nerve 7 through 12, we want you to know what they do and their names, as well as whether or not they're sensory or motor, but you don't actually have to be able to identify them on a specimen. So if you watch the video that our lab coordinator, Jen Thornsberry, has created, she points out the locations of these first 6 cranial nerves. And then we don't ask that you know the locations or be able to identify this next 6, the numbers 7 through 12, because those ones are sometimes harder to identify. Sometimes when the brain is removed from the animal, we lose a good bit of them. So I'm going to flip to the next slide now. to show you, this is no longer a diagram like the last slide was. It's going to give you a little bit better image of what you should expect to see on the actual brain. But again, I want to refer you to the dissection video that Jen put together for you. Okay, so this is, again, this is the olfactory nerve. It's going to be the most towards the front. Okay, most towards the front of the brain. That's nerve 1. The optic nerve would be here, connected to the eyes. Again, some of it just gets ripped off when the specimen is removed from when the brain is removed from the animal. Okay. The oculomotor nerve is here. That's nerve 3. Trochlear is nerve 4. Comes out, like I said, a little bit more on the sides. Trigeminal is nerve 5. Again, that's on the sides. Nerve 6 is abducens, and that is more on the midline here between the pons and the medulla. Two different like structures here on the ventral side of the brain. Okay. So now I'm going to take us to the next slide, where this is just a list of all of the nerves. So far, we've talked a little bit about the functions of the first few. I think I had left off with trochlear, nerve 4. This is also a motor nerve, like nerve 3. And it also allows for movement of the eye. Okay, so it's a motor nerve. It innervates the superior oblique muscles, which allow us to move the eye up, down, and outward. Okay. And then nerve 5, the trigeminal nerve, is a mixed, has sensory and motor information that travel in that nerve. Okay. So it's got, it's both sensory and motor. And for sensory, it helps us to detect touch, temperature, and pain on the face. Okay. In the mouth, teeth, nasal cavity, are all in that area. Okay. And then it also conveys some motor information to the muscles of the jaw for chewing. Okay. The abducens, which is nerve 6, is a motor nerve, and it controls the lateral rectus muscle to abduct the eye laterally, so away from the nose, outwards. Okay. All right, let's move now to the last 6, which these ones, again, we want you to know what they do. We want you to know if they are sensory, motor, or mixed. And of course, we want you to know their names. Okay. So the nerve 7 is the facial nerve, and it's both sensory and motor. It's mixed. And as far as sensory information, it takes taste and information from the front 2/3 of the tongue and allows your central nervous system to process that. And then the motor information that travels in nerve 7 controls your muscles of your face. Okay, so it allows you to make different expressions, and it controls the secretion of tears and saliva and mucus. Okay. All right, and then nerve 8 is the vestibulocochlear nerve. That is a sensory nerve. Okay. And it is responsible for hearing and equilibrium. So in your inner ear, you have different anatomy that we'll look at later in this PowerPoint. And that processes not just hearing, okay, not just the auditory input, but also information about balance and your location in space, movement of your head. Okay, so all of that is information that is important to our central nervous system, where we are in space, how we're moving. We need to maintain equilibrium physically. So the vestibulocochlear nerve is the nerve that processes that information, allows our central nervous system to comprehend that. Okay. All right, nerve 9 is the glossopharyngeal nerve. It's both sensory and motor. For sensory, it controls some information about taste. Okay, so nerve 7, remember, also did that, had taste from the anterior 2/3 of the tongue. The remainder is processed in nerve 9, the glossopharyngeal. And then it also allows for other information from the tongue. So touch, pressure, pain, temperature, sensations from the remainder of the tongue. And then it also processes some sensory information from areas including the pharynx, tonsils, and middle ear. Okay. And then it will also be used to detect blood pressure from the carotid artery. So it allows our central nervous system to respond to changes in our blood pressure. Nerve 10 is the vagus nerve, and it is also both. It has sensory information, including taste, hunger, and fullness. And then for motor information, it controls the muscles that are required for swallowing, talking, and then also um, controls a response to changes in heart rate and blood pressure. Okay, so it allows for decreased heart rate and blood pressure um, if other parts of the body are sending information to the central nervous system that would uh, encourage a change in heart rate and blood pressure. Okay, and then nerve 11 is the accessory. That's a motor nerve. It controls the muscles of the head, neck, and shoulders. Okay. It's required for rotating the head, shrugging the shoulders, movements of that kind. Okay. And then nerve 12 is hypoglossal. That's a motor nerve, and it controls muscles of the tongue required for moving your tongue around and allows us to move food to the back of our mouth. Okay. All right. Now, like I said, you are responsible for knowing the names of each of these nerves, which number corresponds to them, whether they're sensory, motor, or both, and the functions. Okay. So for the names and for the nerve type, whether it's sensory, motor, or both, there are lots of mnemonics. Most of them are dirty, but this one is relatively G-rated. So I'm going to give you a few mnemonics just to help you remember that. Okay. So for what the actual nerve name is, obviously this will be nerve 1 through 12. You can remember a phrase, a mnemonic. So this one is, on occasion, our trusty truck acts funny. Very good vehicle anyhow. Okay, so the first letter, O, corresponds to the first letter of the nerve. Okay, so if you can remember like a little phrase like that, sometimes it helps you to then remember the first letter and then you can get the rest of it. Okay. And then the same thing can be done for whether or not the nerve is sensory, motor, or both. Okay. So for this one, the olfactory nerve is a sensory nerve. So sensory starts with S. We make a mnemonic. Some say money, marry money, but my brother says big brains matter more. Okay. All right. So again, just to help you out when you're studying, okay, you can remember the mnemonics. All right. So we are going to now take a look at the five senses and the receptors for some of them and how the information from the environment is actually processed then for some of these senses. So five senses are olfaction, vision, gustation, audition, and somatosensory. Okay, big words for smell, olfaction, vision, obviously. Gustation is taste, audition is hearing, and somatosensory is touch, temperature, pain, stuff like that. Okay. All right, so they use different types of receptors also. So there are chemoreceptors for olfaction, for smell. Chemoreceptors sense chemicals. And in this case, we're talking about odorants. So little odorants in the air are little chemicals that our olfactory bulb, which is at the end of the olfactory nerve, can process, okay, by means of olfactory receptors that are on specifically olfactory receptor cells. All right, so chemoreceptors are present for the sense of olfaction, the sense of smell, as well as for gustation, our sense of taste. Okay, so again, there are different tastants, little chemicals, which then can be absorbed and through chemoreceptors translate that information about the tastant, whether it's salty, sour, sweet, bitter, umami, which is a savory tastant, to our central nervous system. Okay. And then electromagnetic receptors are responsible for our sense of vision. Photoreceptors, which include the rods and cones, are a type of electromagnetic receptor. Okay. Mechanoreceptors are used in hearing, as well as our sense of touch, our ability to detect pressure. Okay. So mechanoreceptors, again, sense a change in movement or pressure, and we're going to take a look at this for audition in the next couple slides, how we hear based on the movement of fluid and how that fluid movement can be detected by mechanoreceptors. Okay. And then the same type of receptor is used to detect pressure on other parts of our body. Thermoreceptors are responsible for detecting temperature, and nociceptors are the receptors that are responsible for detecting pain. All right, so let's move to audition. Okay, so this is the first sensory system we're going to look at. You're probably familiar by this point with the fact that sound waves are responsible for transmitting the sounds that we hear. Okay, so sound waves moving through the air transmit information about pitch and volume. Okay, so these are various sound waves. Okay, so if you'll notice, the peak here is not as high as the peak on this sound wave. Okay, so that's going to be a quieter volume versus a louder volume. And then if you look at the bottom picture here, the pitch changes as well with the frequency of the wave, how rapidly it peaks and dips. So if it has a slower curvature, a longer curvature, then it's going to have the sound is going to be detected as a deeper pitch. And if it has a quicker curvature, then it's going to be higher pitch. Okay. So all these sound waves are detected by our outer ear. Okay, so the outer ear includes the pinna, which is, the outer portion of our ear here. And then the sound waves are amplified as they move into the auditory canal. So all of that information is sort of funneled in there. And so we can amplify the noise. And they hit the tympanic membrane, which is a little membrane right here. Okay, right at the junction between the outer ear and the middle ear. Okay. So let's move to the next slide to take a closer look at the middle ear then. So we got the pinna, the auditory canal, leading to the tympanic membrane. Okay. And then again, you've probably heard of the three bones that are in the middle ear: the malleus, the incus, and the stapes. Okay, so these three bones, as the sound waves hit the tympanic membrane, these bones are then moved. Okay, so that little pressure on the membrane will move the malleus, the incus, and the stapes, which then moves another membrane here, which is called the oval window. Okay, so it puts a little pressure on the oval window, and then we move to processing by the inner ear. Okay, so the inner ear, it starts with the oval window here, and it will convey that vibration, that information about the sound waves, that vibration to the cochlea. Okay, so the cochlea is this little sort of seashell, spiral shaped structure here. And it is then connected to the vestibulocochlear nerve, nerve 8. Okay, so information that's processed here in the cochlea will be taken through nerve 8 to the auditory cortex of the brain. So let's move to our next slide to look at the inner ear a little bit more closely. So here again is our cochlea. Okay, and if you were to cut a section out of the cochlea and take a look at it from the side, there's a little cut in there. This is what it looks like. You would notice there are various canals in here that hold fluid. Okay, so remember we had the sound wave amplified and exert some pressure on the membranes and the bones, which then ultimately is conveying a movement to the oval window. Okay, and as the oval window butts up against the cochlea, then that vibration is transferred to a movement of the fluid inside the canals, so the vestibular canal and the tympanic canal. All right, so that's inside the cochlea, and the movement of that fluid then in these canals is detected by special hair cells, which are located here in the cochlear duct. Okay, so we're gonna look down below now to see an enlargement of the hair cells. And these hair cells here, these are detecting movement. They have mechanoreceptors, which detect movement of that fluid. that has now been moved by vibrations that traveled through the outer and the middle ear. Okay. So as that happens, then the hair cells move and activate the hair cells. Excuse me, they activate a signal which travels through the auditory uh, nerve, so vestibular cochlear nerve, nerve 8, and that is then taken to the auditory cortex of the brain. Okay, so that is how audition works. It's using mechanoreceptors which detect a change in movement or pressure. Okay. So in the lab this week, we're going to have you look at how hearing works and how it might work if there were a problem with processing auditory information. So there are two different types of deficits that can take place with hearing. One is a conductive hearing loss, which is damage or malfunction in the outer or the middle ear. And then the other, the second type of deficit, is called a sensory neural hearing loss. Okay, and that affects the inner ear. So if it refers to some type of obstruction or damage to the outer or the middle ear, then we call it a conductive hearing loss. If it's the cochlea and further down the line, then it's a sensory neural hearing loss. Okay, and there are two tests that we're going to have you guys do in lab using tuning forks. And these are the Weber and the Rene test. And this helps to localize which part of the ear is the damaged portion. So obviously if a clinician were trying to treat a patient for a hearing loss problem, they would want to know where the problem was. Okay, and so there are two different tests that we will use to look at that. The first, like I said, is the Weber test. So I'll mention here that sound waves can travel through the air, right? And they would be absorbed by our outer ear, or they can actually be conducted through bone vibration. So if you were to place a tuning fork on bone of your head, either like on your forehead or in the middle of your skull, that vibration of the tuning fork after it was activated would be conveyed through the bones of your head to the inner ear. Okay, so we're going to examine whether or not hearing loss is a conductive hearing problem or a sensory neural hearing problem by our ability to sense vibrations through the air versus through the bone. Okay, so if you look at this first picture here, if a person had normal hearing, no deficits, no problems, what you should see then is that if the tuning fork were activated and placed on the bone of the head, then you would hear pretty much the same on both sides. So you shouldn't have one ear sensing more noise than the other. It should be pretty much the same, okay, if it's placed in the center of your skull. If you do hear the noise, perceive the noise as being louder on one side than the other, this is called lateralization, okay? And lateralization in this test can indicate either ipsilateral, which means same side, conductive hearing loss, or contralateral sensory neural hearing loss. So for instance, if you have a bad ear, you have something wrong, you know you have something wrong on one ear, and you sense that when doing the Weber test that that ear sounds like it's louder, then you might have a conductive hearing loss on that side. Okay, so that's again a hearing loss that is affecting the outer or the middle ear. Okay, whereas if your bad ear that you know from experience is the one that is not really working, is on one side, and then you have lateralization, so you sense the sound as being stronger on the other side, then what that indicates is the bad side has a sensory neural hearing loss. So that's indicated by this last picture here, where this stronger bold red line is indicating that there's lateralization to the opposite side, the contralateral side. Okay, so what that means then is that you've got a sensory neural hearing problem on this side. Okay, so this is one test that's used to determine which part of the ear is affected in someone who's having hearing problems. Like I said, the other test is called the Rene test. And this is also using a tuning fork. Okay, so you activate a tuning fork and then you place it on the mastoid bone, which is the bone behind the ear. Okay. And what you would normally expect if you had no hearing loss problem is that you would hear the sound when placed on your bone for a time period, and then if you remove it from the bone and place it outside the ear, so in the air right outside the ear, you should be able to hear it longer. So place it on your bone, you stop sensing any noise after say 10 seconds, then you should be able to move it to the air outside your ear and continue then to hear the noise again for another say 10 seconds. Okay, so what this is saying is that with no hearing deficits, the person should be able to hear the tuning fork via air conduction twice as long as via bone conduction. Okay, and that's shown in the top picture here. So they move the tuning fork from the bone to the air, and the individual could again sense the noise. Now, this is again if the person has normal hearing. So hopefully all of you, when you do this in lab, have normal hearing, and you'll be able to hear the noise longer via air conduction than via bone conduction. Okay, but if bone conduction is perceived as better than air conduction, so if you don't hear it longer via air conduction than bone conduction, then you can conclude there's a conductive hearing loss problem. So something wrong with the outer or the middle ear. Okay, and that's shown here. Okay, so if you still hear it, but then you move it, if you still hear it through the bone, then you move it to the air and you don't sense the noise any longer, then you might have either some obstruction to the outer ear or damage to the middle ear. Okay, something is wrong there. If you don't hear anything for either one, then you might have a sensory neural hearing problem. So if hearing's poor for both bone conduction and air conduction, then you might have a sensory neural hearing problem, something wrong with the inner ear. Okay. All right. And as I had mentioned a few slides back, nerve 8, the vestibulocochlear nerve, senses not just that auditory information, but also information about our equilibrium. So our position, especially our head position in space. Okay. So this is done through semicircular canals, which are these 3 little partial rings that are located just above the cochlea. Okay. They also have hair cells, and this gelatinous mixture called a cupula, which is located in a canal with fluid. So in these canals that have fluid moving through them. Okay, and as you move your head in one of the planes, so left, right, up, down, tilted, then each of these semicircular canals has the ability to respond to one of those movements, okay? And the fluid in the canal will move, and that will displace the cupula, which is attached to the hairs on the hair cells. And then that information about the movement of the head is able to be transmitted to the brain, okay? Through, again, nerve 8, okay? All right, so that wraps up the description of the sensory information that is taken from nerve 8. You'll do a couple exercises in lab that takes a look at that also, but that's the end of that. So now let's talk about temperature. All right, so temperature is detected by thermoreceptors and then processed in the posterior hypothalamus, which is part of the brain below the thalamus. All right, and so temperature is obviously very important to detect, but we can become adjusted to a temperature. We can sort of acclimate to a temperature. That's why I often feel like in the spring, 60 degrees is like beautiful. It's so nice and warm, even though in the fall to me, I'm like, oh, it's getting kind of cold, you know? So this is what's called as acclimation or acclimatization. All right, so change in temperature occurs over a period of time, we can adapt to it. So we're going to have you do an experiment in lab that examines this, our ability to respond to this, okay? And it basically involves putting your hands in some really cold or warm water, and then seeing which adapts faster. And then once you move to a room temperature water, which one seems to be what you feel in response. So if you had your hand in cold water and put it in room temperature water, what you might feel. Does it feel like it's room temperature, or does it feel really warm? And then if you had it in warm water and you put it in room temperature, does it feel cold or does it feel lukewarm? Okay, so we're going to have you guys do an exercise in lab that examines that. And of course, that's regulated by thermoreceptors. Okay. Pain is processed by nociceptors. And the concept we are going to have you guys look at in lab for this is referred pain, which is pain that's perceived in an area of the body that is not actually being damaged. Okay, so the classic example of this is that when a person has a heart attack, it's often realized, sensed by a pain in their left arm or shoulder, right? Even though there's nothing actually wrong with their left arm, the damage is actually taking place in or around the heart. It's perceived as pain in their arm. And the reason that this concept occurs is that when there's a nerve that crosses over multiple regions of the body, so information that is conveyed from the arm might also convey information about the heart. And so the same nerve is processing information from more than one area. And so that response, that sensation, that perceived pain is unclear to your central nervous system where it's at, where the damage is actually occurring. Okay, so for this exercise in lab, we're going to do an experiment. You're going to put your elbow in icy water, cold. It's uncomfortable. But after a period of time, what you might feel is a change in location of the sensation. So it's, again, it would be affecting your elbow because your elbow is in the icy water. But what people tend to feel then is pain in their last two fingers, so your pinky and ring finger. And that is because your ulnar nerve, which is shown here in yellow, is innervating the whole length of the arm on that side. Okay, so the pain of putting your elbow in icy water is sort of sensed in your fingers, which of course are not the ones being affected. But if you do this exercise, hopefully you'll understand and be able to experience this concept of referred pain. All right, so let's turn our attention to vision now for just a few minutes. We do have a couple experiments in lab we want you to do to examine our visual sensory information. But first, we want you to know a little bit of anatomy about the eye. Okay. So the outer portion of the eye is the sclera. That's the white part. Okay. And then towards the front, that becomes the cornea, which is transparent and covers the iris and the pupil. Okay. So just underneath that then is another layer called the choroid, which at the front becomes the iris. Okay, and the iris is the colored part of your eye. And then in the very middle of the iris is the pupil, which allows for various amounts of light to enter the eye. And of course, the movement of muscles that control the iris allows for that pupil to be smaller or larger and allow less or more light into the eye. Okay. And then behind the pupil is a lens, and this lens is connected via suspensory ligaments and some muscles, which will control whether or not the lens is bulged or thin. Okay. And this allows for a change in focusing of the light that's entering as it hits then the retina. Okay, so the retina is the inner layer, and that's where all our photoreceptors are, our rods and cones. Okay, so you've got all the photoreceptors in sort of the back in the retina, and the light needs to be focused on that retina to allow for our body to process visual information. Okay. All right. Once it hits the retina, then that information is conveyed through the optic nerve, which again is nerve 2. It's a sensory nerve, okay, to the brain. And I'll mention that there is a point where the optic nerve hits the retina, and this is called the optic disc. And at this point, there are no actual rods and cones. There are no photoreceptors at that small part where the nerve connects to the retina. There's nothing that can sense there. So you actually have a little blind spot, which um, will have you evaluate through one of the experiments that you do in lab. We'll have you be able to see that. All right. So I think I've hit on all of the anatomy here. Let's move to the next slide. I wanted you to look at how changes in the lens can change focusing of light. So for instance, if you are looking at something that's far away, far vision, okay, so looking far away, then the lens, which is indicated by this little oval here, is going to be thinner, okay? It's going to be thinned out to allow for focusing that light on the retina, which is back here, okay? This is obviously the front of your eye. Light's coming in this way. And then if you change and look at something near to yourself, closer to you, then this is creating a bulge. Your body will then respond to create a bulge in the lens, which allows for a greater focusing of the light to, again, hit the retina and allow us to see clearly. Okay. And this change in the lens is mediated by ciliary muscles, which are these things in red here, just shown in the diagram in red, ciliary muscles, and suspensory ligaments. So when you're looking far away, the ciliary muscles relax and the suspensory ligaments are pulled tight. And the border of the choroid, which again is this little section here that's shown in red, okay, the border of the choroid moves away from the lens, and that allows the lens to be thinned out, pulled, okay? So we're looking at sort of a movement of that choroid away and pulling then of the lens to thin it out. Okay. And then again, if you're changing to something that is closer to you, to view something closer to you, then this is ciliary muscles are contracting, suspensory ligaments sort of slacken, and then the border of the choroid is pulled towards the lens, and that again, that pulling towards the lens is allowing the lens to bulge out a little bit, okay? And that controls then the focusing of the light on the retina, okay? All right, so that's called the accommodation reflex, the changing when you're viewing from far to near. That's called the accommodation reflex. I don't think we're actually going to have you evaluate that in lab this year because it requires other people to be closer to you. But you should be aware of the fact that the lens does change shape in order to focus light appropriately from different distances. Okay. All right, so this week in lab, we have a sheep brain for you to look at. And I do want you to look at that video that Jen created for you guys. We want you to identify physically cranial nerves 1 through 6, and then know the functions and the names, whether they're sensory, motor, or both, for all 12 cranial nerves, okay? And then we're gonna have you guys look through, work through the lab manual activities, which explore sensory input of various kinds, and know, again, be able to identify which nerve is responsible for conducting which of these sensory inputs, okay? And then the assignments for this week, I believe, are all McGraw-Hill assignments that are already posted on Canvas. They might not be available though until the beginning of this week's lab. Okay, so if I post this video, they might not be available till Monday or Tuesday. Okay. All right, so I think that wraps up this week's pre-lab. Hope you guys are having a good weekend. I hope to see you soon.